Video we'll talk about the ventricles and cerebrospinal fluid. So let's have a quick look at the diagram here. We can see that there's two views, a side view and a head-on view. Let's look from the side view. Here is the ventricular network, and we have two ventricles. We have one lateral ventricle, and behind it we have a second lateral ventricle, and they occupy, one occupies each of the hemispheres, and you can see that horn shape to it. Now, these are ventricles 1 and 2, but we just call them the lateral ventricles. And so here's one ventricle, here's another. You can see the shape of it, each one, and the contents from those will then enter this space right here, which we will call the third ventricle. And from the third ventricle, cerebrospinal fluid will flow down this canal, which we call the cerebral aqueduct. And from the cerebral aqueduct, it opens this wide area here. And this is the fourth ventricle. So we've gone from the two laterals into the third ventricle, down the cerebral aqueduct, into the fourth ventricle. Now from here, cerebrospinal fluid has a few options. There are two lateral. There's a lateral aperture, and there's another lateral aperture here. Then there's a median aperture. So these are places where the cerebrospinal fluid can flow through and out. So when it flows from here, it'll flow over the top surface of the brain in that subarachnoid space. And eventually, as we saw in the last video, gets picked up by the sagittal sinus, because CSF comes from the blood and is returned to the blood. It's basically a filtrate. There's another route, is that a little bit of the fluid can continue on down in the spinal cord in the central canal. Let's just look at it from the side. Once again, we can see the lateral ventricles, one in each hemisphere. They meet into the third ventricle. From the third ventricle, the cerebral aqueduct takes it down to the fourth ventricle. And as I said, from there are the apertures, the one median and two lateral, which will take the fluid up and over the brain. Now, let's just talk a bit about it. We'll just mention what we just said, and that is the ventricles. There are four fluid-filled chambers in the brain. There's two lateral, one in each cerebral hemisphere, and then we also have the third, which is a narrow space just beneath the corpus callosum. We have the fourth ventricle, which is a small triangular chamber, and it's located between the pons and the cerebellum. And they are connected. Third and fourth are connected by that cerebral aqueduct. So let's talk a bit about the connections. Is that the ventricles are connected, and we have the interventricular foramina, and that's a pore that connects the lateral ventricles with the third ventricle. Then we have the cerebral aqueduct, and that's a tube that basically connects the third ventricle to the fourth ventricle. And as I said, we also have the continuation of the through the central canal, which basically runs through the center of the spinal cord. Here we can just see in section. Let's spend a few minutes here. I'm just going to walk down through some of the knowledge we've gained so far in the previous video. And here you can actually see there's the gray matter. In the last video we mentioned the gray matter is on the outside. Here you can see the extensive white fiber tracks that are running through here. And as I said in the last video, you can see there's dark islands of gray matter scattered deep into the brain. So that rule that in the brain, gray matter is really at the periphery, white matter occupies most of the interior, and then there's islands of gray matter that are buried in here. They're the various nuclei. Now to get back to the ventricles, here we can see part of a lateral ventricle and part of a lateral ventricle. Because remember, we're looking at a structure that goes kind of up and over like this. And that's why we're seeing part of it here, part of it here. So that's why we're seeing these bits right there. Here is actually a good view of something called the choroid plexus. part of the choroid plexus, and we'll talk about what that is in a few minutes. So, what is cerebrospinal fluid? Cerebrospinal fluid is also known as CSF. It's clear, it's colorless liquid. It's a derivative of blood, and of course it fills the ventricles and all the canals of the central nervous system. And as we mentioned earlier, it leaves through the apertures and will bathe the brain on its external surface. And CSF begins with the filtration of blood plasma through the specialized capillaries of the brain. And we just saw in the previous, I highlighted the choroid plexus. Go back, zip back if you need to see it. And the choroid plexus is basically this mass of blood capillaries on the floor of each ventricle. And it's through these specialized capillaries that we get basically a leakage of fluid and certain things. So CSF isn't the same as blood, but it's derived from blood. It has some of the components of it. There's also the ependyma. Remember, they're the type of neuroglial that lines the ventricles, and they cover the choroid plexus. Now, what's their job? They do a few things, and what they really are involved is that they help to modify, just changing colors here, they modify the filtrate. So when I compare CSF and blood, I notice that they're not quite the same. CSF has more sodium and chloride, less potassium, also glucose, and very little protein is found in there. So it's not quite the same as blood, but it is a filtrate of blood. And CSF is made continuously. As CSF is made, it flows from say the lateral ventricles to the third to the fourth, bathes over the brain, and goes back into the sagittal sinus, that blood cavity. And so it's made continuously. So I'm constantly making new CSF, and I'm reabsorbing CSF that's basically flowed through that route. So CSF continuously flows through there. Now, what's driving it? Well, of course, its own pressure, because if I'm making new stuff, it's going to push the older stuff ahead of it further on, and I'm going to build up pressure. And remember, things go from high pressure zones to low pressure zones. Also, the ependymal cells have cilia, which help to basically push it along. And there's another thing, is that actually the pulsation of the brain with each heartbeat also has a role. And as we said, the root is, it's secreted in the lateral ventricles. There's a few other places where it's also made. It's not just the lateral ventricles. It passes through the intervertebral foramina into the third ventricle, down the cerebral aqueduct to the fourth, and the third and fourth ventricles can add more CSF along the way. Now, all the CSF basically will escape through the three pores that lead into the subarachnoid space, and also the spinal cord surface. As we saw earlier, we have the median aperture and the two lateral apertures. And CSF gets reabsorbed by the arachnoid granulations, and that is something that we had a look at, the picture of it, when we looked at meninges in the previous video. So have a look at your book if you need sort of get that visual representation, or go back to the previous video. And these look like cauliflower or broccoli or whatever works for you. They're these extensions of the arachnoid layer into the blood sinus. So they protrude through the dura mater and into the superior sagittal sinus. And the CSF basically penetrates the walls of the villi and mixes with the blood in the sinus. So I'm able to return that fluid and some of its constituents back into the blood. We can see here on the diagram, this is a side view. You can see the basic flow is that here's the lateral ventricle. You can see here the choroid plexus. CSF is made. It flows down into the third ventricle. From the third down to the cerebral aqueduct. You can see this is the fourth ventricle. And also we have some chord plexus here too. And you can see where it's located. The fourth ventricle, that's the fourth ventricle in this region, is between the brain stem and the cerebellum right there. And then the fluid will leave through the apertures, the three apertures that we talked about. It can surround the spinal cord. It can also flow up and over the brain, the surface of the brain in the subarachnoid space. And then in mid-sagittal, the CSF can enter the blood in that sagittal space. And you can also see that the CSF will flow right over the whole surface of the brain, even kind of come up over the outside in this direction. And the end result is that, I'm making CSF throughout the ventricles. It's flowing in a given direction based on pressure and the action of the ependymal cells and their cilia, and the fluid gets returned eventually to that mid-sagittal sinus. Now, what are the functions of CSF? If we go all this trouble making it, what does it do for us? Well, it gives us buoyancy. Think about the brain. The brain does have some mass, and if the brain didn't have sort of like bubble wrap or like a little, water bed underneath it, the weight of the brain would be sitting on the hard bony tissues of the bottom part of the inside of the skull. And this could potentially do damage. It could actually destroy the nervous tissue. So it acts as that sort of bubble wrap water layer to separate the brain from the actual bony tissue. So yeah, the brain is quite big, and it does have a significant amount of weight, and if it rests on the floor, we could have issues with basically destroying the nervous tissue. That is not good, as you can imagine. The next thing is protection. It also protects the brain from striking the cranium when the head is jolted. So, quick motions of the head. The brain is independent from the actual bony tissue, although it's quite close to it. That water layer just gives you a little bit of protection. Now, it's not fabulous protection. It's not designed for you to fall off your bike without a bike helmet and hit your head, or sadly something like shaken child syndrome, that you can still get injuries to the brain. Also, it provides a chemical stability because we're very careful as to what we allow to enter the CSF. As you can imagine, the neurons are really dependent upon a certain environment of chemicals outside and inside them to maintain their ability to do action potentials and basically do their daily work. So we have to be very careful as to what will leave the blood and enter the CSF. Also, it plays a role in removing metabolic waste from the nervous tissue, so it also does homeostasis and remove metabolic waste. Now, a little bit about the blood supply to the brain. The brain only is about 2% of the adult body weight, but look at this, it receives 15% of the blood. And not surprisingly, because these are very metabolically active cells, and you can't afford to have them go offline. And the neurons have very high demand for ATP, therefore oxygen and glucose have to keep coming in. To give you an idea, I wouldn't hold you to these numbers, but to illustrate it, that even just 10 seconds interruption in blood supply, you can end up with loss of consciousness. Within 1 or 2 minutes, you get significant impairment of neural function. And within about 4 minutes, unless under extreme circumstances like very reduced body temperature or something like that may mitigate this a little bit, you're looking at maybe 4 minutes until you start to see irreversible brain damage. Now, there's a blood barrier system that regulates what can get from the bloodstream into the brain. And we got to be careful we don't allow harmful agents within there. So this blood barrier brain system is selectively permeable. So what can cross? So the things that get the thumbs up and are able to cross, well, water, glucose, lipid-soluble substances like oxygen, carbon dioxide. By the way, alcohol is very lipid-soluble, so it crosses, and thus the effect of alcohol. Caffeine, nicotine, and not surprisingly, anesthetics. That if you want to use a chemical as an anesthetic, then it should probably be able to cross the blood-brain barrier, and they do, right? Now, we're going to talk about this issue, is that this really can limit therapeutics that you're trying to target to the brain tissue. It's slightly permeable to sodium, potassium, chloride, waste products, urea, creatine. And 2 points of entry have to be guarded, and that is the blood capillaries throughout the brain tissue, I should say, and the capillaries of the choroid plexus, because this is the two places the brain interacts with it. So at the blood capillaries, the brain is protected by the blood-brain barrier. And let's just underline that to emphasize it. So it's the blood-brain barrier. And this is really, we're really looking at tight junctions. Tight junctions play a big role here. So tight junctions between the endothelial cells. And during development, what happens is the astrocytes, as one of their jobs, reaches out, it contacts the capillaries with their perivascular feet. If you're not sure what these look like, go back to previous chapter where we talked about glial cells. And what they do is they tell the endothelial cells to form tight junctions that completely seal off the gaps between them. So now anything that wants to leave the blood cannot squeeze between the endothelial cells that make up the wall of the capillary. The materials actually have to enter the cells, cross the cells, and leave them. So we're talking about transcytosis. So it gives you much more control. And anything leaving the blood must basically pass through the cells and not the gaps between them. This allows the endothelial cells to be selective, unlike if you had simple gaps where as long as you're small enough, have the right chemical nature, you could go right through. So this hopefully eliminates harmful substances from getting into the brain tissue. Now, the other place where we have to worry about the interaction between blood vessels and potentially brain tissue is the choroid plexus. And remember, this is where we make CSF. And the brain is protected by the blood-CSF barrier. And once again, we have tight junctions formed between the ependymal cells. Now, let's talk a bit about the blood supply and the blood-brain barrier. Now, typically it's just called the blood-brain barrier or blood-brain system, BBS if you want. Now, most of the time this does us a favor, but there are times where in terms of pathology it becomes an issue in that it's an obstacle for delivering things such as antibiotics or anti-cancer drugs or cancer treatment drugs to the brain, because many pharmaceuticals cannot easily cross that system and actually enter the brain itself. Now, trauma and infection can actually damage the BBS and allow pathogens to enter. Now, there's certain places called the CVOs, which is circumventricular organs. And these are in the third and fourth ventricle where there's no barrier. Now, we'll talk more about these when we get to the respiratory system. These are very specialized areas in the brain where the brain does quality control, looking at blood gases to make sure that the brain is getting adequate amounts of oxygen and isn't having a buildup of CO2. Now, the discussion would be large to go into great details, but we will come back on it. But the short message is, in these particular areas, what we're looking at is we're trying to measure things. So it actually allows blood to direct access to the brain. Now, this is a bit of a trade-off that we're doing this. But what it does, it allows the brain to monitor and respond to fluctuations in things of, now look how important these are. Blood glucose, that is something that the brain would want to know. If blood glucose is going down, it would want to know about that because it's trying to look after itself. The next one is an interesting one, and it's pH. Now, pH, we're going to talk a lot about in the next course. And if you see pH here, what we're really thinking of, it's a surrogate for CO2. So by measuring pH, the brain has a way to determine CO2 levels, which tells it some very important things about blood gases. Are you breathing fast enough? Are you breathing deep enough to bring in oxygen and to get rid of CO2? It also can measure osmolarity, and there's a whole number of other variables. Now, unfortunately, some pathogens can actually use this route. So the human immunodeficiency virus, HIV, can actually use this route to invade the brain. Now, I'm not going to say too much here about strokes, but since we are talking about it, so a stroke, often referred to as a cerebrovascular accident, a CVA, this is where you stop or interrupt blood supply, which can quickly cause death of brain tissue. Can occur at any age, but typically, 60-some percent, say about 65, 66%, are those over the age of 65. Also, the second greatest cause of death after coronary artery disease, so that's quite significant. And it's not unusual that about 50% of stroke patients live less than 1 year post-stroke. That often it is very damaging, and often with strokes it's about location and extent. And just want to point out, we use the word stroke, but there's two big different processes that are quite different from each other. That we can have hemorrhagic stroke, and the hemorrhagic, of course, refers to bleeding. So this could be a rupture of a cerebral or a subarachnoid blood vessel. And in that case, you basically get a loss of blood flow, but you're also getting blood that is now pouring into nervous tissue itself, which can be detrimental to the tissue, and you can get a pressure buildup, etc. The other one is obstruction of blood vessels, and this would be something such as a blood clot, a thrombosis, or lipid deposit, which is like an atherosclerosis, or a breakup, a chunk of lipid sort of composite breaking off in another blood vessel and traveling up here and basically obstructing. So, and these two types of strokes are very different creatures in terms of how they're dealt with, but the end result is both of them disrupt blood supply. Okay, we're going to end it there, and next video we're going to pick it up and we're going to start to walk our way up the lower part of the brain. We're going to work our way through the medulla, the pons, into the midbrain, and continue up from there.